Good afternoon. We are still having a number of people logging on, but we will go ahead and start today's event. My name is Dr. Alastair Cribb. I'm Dean at the Tufts University Cummings School of Veterinary Medicine. Thank you for joining us all today. This is the third in a four-part Tufts University faculty webinar series called A Force for Good, Tufts on the COVID-19 pandemic. Just a couple of housekeeping things to start us off. Please note that you are all muted. Everyone listening is muted, and the chat feature has been turned off. If you want to send in a question during or at the end of the seminar, we'll do all the questions after the presentation. You do that through the Q&A on your Zoom. So to get to the Q&A, you just move your pointer to the bottom, and the band will come up, and you can click on the Q&A. I'll then read out the questions at the end, and Dr. Runstadler will answer them. And we'd like to thank Dr. Runstadler for joining us today to talk on your pets and COVID-19 and tell you a little bit about the research that he is doing. Dr. Runstadler is a New Hampshire native. He did his Bachelor of Science degree at Stanford University. He followed that up with a DVM degree at UC Davis and then a PhD in immunogenetics. Subsequent to that, he joined the University of Alaska Fairbanks as an assistant professor, and he was a member of the Institute of Arctic Biology and then ultimately the director of the Alaska Zoonotic Disease Center. Following his time at the University of Alaska Fairbanks, he moved to the Massachusetts Institute of Technology, and he joined Cummings School of Veterinary Medicine in 2017 as a professor in our Department of Infectious Disease and Global Health. He is a virologist and an immunologist. He does a lot of research on avian influenza, and he's really interested in emerging diseases. So when COVID-19 started to occur in the United States, he immediately started doing research in that area, and that is what he's going to share with us today. So I'll turn it over now to Dr. Runstadler.
Thank you, Dean Cribb, and thanks everybody for joining in today. So I'm going to talk a little bit about a particular study that we've started to study COVID-19 and possible transmission between humans and pets. And as Dean Cribb mentioned, my group has been working on influenza virus for a long time, which is from the standpoint of emergence of viruses in the human populations, and the potential for that to happen is a very similar situation as with COVID-19. And so when COVID-19 was clearly an issue, even at the beginning of the year in January, my group started thinking about ways that we could contribute to perhaps alleviating or at least studying this epidemic and now obviously pandemic event that's affecting everybody. And one of the ways that we understood that we could do that based on our experience with influenza virus was to understand if this virus has the potential to move back and forth between animal hosts and human hosts. And so we began a study very early on, basically to sample animals that were in close contact with humans and to see and to collect some data on how much infection or transmission was actually moving between humans and their animal contacts. So before I'm just going to tell you a little bit about that study and some of our preliminary results and kind of where we want to go with that, and then happy to answer any questions that come through, of course. But before I start, I just want to thank a lot of people and a lot of groups, and this is a growing list, and this one is actually probably a little dated, but we started out, as I'm going to tell you, sampling animals that are coming through the Cummings Veterinary Medical Center at the veterinary school out at Tufts, and we've been expanding a bit ever since. And the group of contributors to this study includes a lot of different people, including some additional veterinary hospitals, as well as wildlife and zoo and shelter groups as well. And so we're really grateful to the enthusiasm and help that everybody has given to getting this study going and to continuing it. So, I think we asked a question that a lot of people were asking really early on at the beginning of the year, as it was clear that the epidemic in China was growing rapidly and likely to go beyond that region and worldwide. And that is, do we expect that animals might get infected? If there's an emerging virus that's spreading rapidly in the human population, are animals at risk? And there was at least a couple reasons to think that that could happen. One of those is that this virus and some of the genomes that were sequenced from early on in China revealed a really high degree of similarity with viruses that have been found in bat populations in the same region. And in fact, some of the epidemiology early on pinpointed some live animal markets as possible sites of emergence of that virus into the human population. So we kind of know that this virus came out of animal groups, whether they were intermediate hosts in contact with humans or a wildlife species. And secondly, we know that this has happened in the past. Coronaviruses are a family of viruses that is found in a wide variety of animal hosts, including most mammalian species. And with the original SARS virus that emerged in 2003, several different animals were implicated in the spread of that virus ultimately into the human population, including the civet cat, which is pictured here in the middle of the screen. So based on our expectations from that background, we initiated this project, which we dubbed the COVERS project, and that stands for Coronavirus Epidemiological Research and Surveillance. And our goal here was several-fold, but one, it was to find out if virus was moving from humans back into animals that were in close human contact. And two, really to find out how much that was happening and if that was a risk or a threat to veterinarians who were caring for those animals. So we started to get that study together, and as we were doing so, some initial reports came out of actual infection being documented in cats and dogs, originally from a dog in Hong Kong, and subsequently reports of cats in Belgium and in the US here. There's only been a handful, but a couple of them are fairly well documented. So it seems like those are instances of transmission of the virus from a human host into an animal. And then probably most sort of sensationalistic, probably everybody heard about the tiger and subsequently some of the lions at the Bronx Zoo that were infected. And this is Nadia, the 4-year-old Malayan tiger that was initially sampled when she was undergoing a veterinary procedure, and that sample was tested for COVID-19 and came back positive. And what was interesting is those big cats at the Bronx Zoo showed some very mild symptoms of a dry cough and respiratory illness, which was similar in some ways to human illness. And they suspect that a zookeeper at the time who was asymptomatic but infected with COVID-19 transmitted the virus to these big cats. So subsequently, a lot of zoos have undertaken precautions to prevent that kind of transmission from taking place. So we began our study with the Foster Hospital for Small Animals, which at the time in late March had reduced their service to emergency and urgent business only. And so we were sampling animals that came through mostly the emergency service. And we were asking people to text or call a hotline number that we set up and we set up a website for the study to explain what we were doing and why. And we started to sample animals that were coming through the foster animal clinic. But our study has sort of some different phases to it. That was the initial phase, to just get things going, collect some data, see if these infections that had been documented in the press were rare events or more common things that we might be more worried about. And so that initial sampling involved those pets that were coming into the veterinary clinic, and we've subsequently expanded to several regional clinics and other facilities, and are still expanding a bit. And so we're collecting, in those cases, a single swab sample from the mouth or the nose of the pet, and putting that in a viral transport media, which we then take to our lab, and we screen using a PCR test for presence of the virus in those samples. And I'll show some of the initial data in just a minute from that study. What we've done since then, in late April and early parts of May, we started a second phase of the study where, through the website mainly, we were recruiting volunteers that were interested in participating and would receive a sampling kit through the mail, which they would then use to sample their pet in the home situation over a period of 2 to 3 weeks. So taking a sample from the pet every 2 or 3 days, so we get multiple samples from that household. And part of the intent here was to start to enroll people and their animals in the study who were in more high-risk situations. So people who either had been diagnosed as COVID positive or knew that they had a close contact who was diagnosed as COVID positive. And then we're now entering what we hope is a third phase of the study in early June here, where we're hoping to add the humans in those home study situations to the sampling, so that if we do detect transmission events, we can look at the virus as it originated in the human host and the virus that was transmitted to the animal, and compare those viruses to see if any important mutations are taking place. Then we're also interested in adding other situations, for instance, at the veterinary school here, where we have animal herds and agricultural herds that also may be undergoing close contact with their human caretakers, and to look and see if there's transmission occurring in those animals. And we're particularly interested to see, again, if transmission occurs, how that transmission might move through a herd of animals in an agricultural situation. So those are sort of 3 phases of the study that we've instituted 2 of them and are beginning the 3rd phase. And then as a sort of 4th phase, what we're going to be adding in over the summer here is to add serology to the study. So in all of those different parts of it, to look for the presence of antibodies that indicate that the host has been infected in the past and generated an immune response to the virus. And so we know that as we're sampling and looking directly for the virus, we're certainly maybe missing cases where the animal had been infected prior to our sampling, or the animal may be infected after our sampling, and we could go back and get a blood sample or possibly another swab sample and look for antibodies. So I'll share some of the initial data that we have from that study. And we've sampled now about probably about 400 animals between the sampling at the clinics and that as a part of the home study. This chart breaks down on the vertical axis the number of animals of each species that we've sampled, and you can see that this is mostly dogs and cats, and that reflects what we mostly see in the veterinary clinic and in the home situation. But there are some exotic species in there, including the ferrets and rabbits, We've sampled a few animals through the large animal service at the hospital, including horses and a pig. And then way down there on the right-hand side, there's a hedgehog. I think that was the first animal that we actually sampled in the Foster Hospital for Small Animals at Tufts. And then we have some interesting wild animal species in there. We're working with the Mystic Aquarium to sample seals that they are getting in for rehabilitation. And those seals, again, are obviously in a very close contact human-animal situation. And then we're also have gotten quite a few samples in collaboration with the Wildlife Service here on the Tufts campus of bats. And bats are in some ways of particular interest because it's believed that this coronavirus and several other coronaviruses may have originated out of bat species. And the bats themselves have several different coronaviruses that infect them. So we think they may be a particularly susceptible host, and we're interested in whether virus can move from humans into bats that are in rehabilitation situations. So we've gotten a number of bats to test. The good news really all the way around is that all of the samples that we've tested thus far, and again, that's about 400 different animals, some of them tested multiple times in that home study situation, all of those samples have been negative for the SARS coronavirus 2, the current virus causing the outbreak of COVID-19. And this chart is shaded, the big negative bar there is shaded according to a questionnaire that we're also administering to those clients that are enrolling in the study that are interested in participating, and ask them some questions about whether they've experienced symptoms of COVID disease, and also about what their contact is with the particular animal that we're sampling. So in a pet situation, you know, do they let the animal sleep with them? Do they kiss the animal? How often is the animal going outside? Those kind of things. And that's for when we do get positive cases. If we do, we can look at some of the behaviors that may be associated with those positive cases. At the time, it looks like that's not going to be as much of a factor as we initially thought it might be. So this does seem, our data is consistent with what we've seen really globally, and that we just have the few handful of reports of virus transmission into animals. And so this looks like it's a rare event, and several other groups are now also sampling animals through either veterinary or agricultural context. And it appears that this is a rare event that virus moves from the human host into an animal host. So just to give you a flavor of the home study, this chart is shaded in the same way, where people that reported COVID exposure, either from themselves being positive or from a close contact being positive, we're screening animals that are much more, that are in more of a high-risk situation in that home study. So more of the people with COVID exposure. And still, all of those samples thus far have been negative. So we hope that this study is actually going to continue. We don't know when the epidemic or the pandemic event will end in the human population, and certainly right now it doesn't appear that that's going to be soon. Obviously, we all hope that the development of vaccine will hasten that in terms of the epidemic, but it's likely that that this virus will continue to circulate in the human population into 2021, and so we're hoping to continue sampling animals, particularly in those high-risk human-animal interactions. And part of the reason why we're interested in doing that is that even if we get a rare transmission of the virus into an animal host, we'd really like to look at what happens to the virus in that situation. And it's thought that the emergence of this coronavirus into the human population was probably due to a single event, perhaps at a wet market selling wild animals, perhaps in another context, but a single event of the transmission into a human host and then onward transmission from that human host. So there's some reason to believe that could happen in the reverse direction. And so we want to be able to monitor that, understand what's happening in those transmission event situations, even if they're rare. And in part, we hope that'll help us be prepared for the next event and the next emergence of a virus, either into animal hosts or into the human population. And I'm just going to end here by saying that we're also using these samples to test for influenza virus, which is a virus that my lab has been studying for quite a while, because we're also in a similar way interested in how influenza viruses may be moving between animals and human hosts. And curiously enough, a couple of those samples from this group have turned up positive for influenza. So we're following those up to understand whether that's a human virus that's in those animals or whether that's a virus that circulates in the animal host. And we hope to have more on that. So I'm going to stop there for now and happy to answer questions either about our study or about the transmission between pets and humans of the virus in general. And look forward to doing that. Dean Cribb.
Yes, thank you very much, Dr. Runstadler. That was great. We do have probably about 20 questions here. Some of them are a little bit interrelated, and so I'm going to paraphrase the questions and make sure that we get the key questions answered. I'm going to start with some of the more technical questions around your particular study, and then we'll move up to some of the broader ones if that works for you, John.
That sounds great. Thanks.
Okay, and I will just, you mentioned the Foster Hospital for Small Animals and our clinic, so I will just say a couple of words about Cummings School of Veterinary Medicine before I get into the detailed questions. And for those of you who don't know, we are located about 35 miles outside Boston. And we, as you have gathered from this presentation, have a strong and active research program in infectious disease and global health. We do a lot of clinical research. We do a lot of neurosciences research. So there's a broad range of research that is relevant to both human health and animal health. John's example of a study here being a good one of something that has relevance and veterinary medicine. We actually have 7 clinics that serve the area, and this is where Dr. Runstadler has been able to collect a number of his samples. And that actually leads into a very short question first, which is, do you secure permission from the clients before collecting the samples?
Yeah, that's an important part, and so part of the challenge of a study like this is in part the doing it, and this was really amplified in our current situation of the human epidemic and the fact that we had to basically shut down most of the clinic business except for emergency business. And some of the procedures in doing that actually make it very difficult for us to contact clients and ask if they're interested in the study in the first place. But yeah, part of doing the sampling here is getting permission from the owner, from the client to do so. And so that involves informing them about the study that we're doing and the sampling that we're doing. We're fortunate in some ways in that this study is a very non-invasive sampling. It's simply essentially taking a Q-tip-like swab and inserting it either into the nose or into the mouth to get a sample from the mucous membranes in those locations.
So another question, actually, we're going to get into some testing here, but could you just clarify what the difference is between SARS coronavirus 2 and COVID-19?
Absolutely. So in the simplest sense, the viruses themselves have names, and we often then associate the disease that's caused by the virus with another name. So SARS-CoV-2 or SARS coronavirus 2 is the name for the virus that is causing the disease COVID-19.
So there's several questions about how you then determine if an animal actually is infected with SARS coronavirus 2. One of the viewers noted that you had on your slides SARS-CoV, RT-PCR, and then you have some other letters behind that. Someone was commenting on the Abbott test and how it's known that that has probably more false negatives than initially thought. So they're wondering, what did those words mean on your slide, and what is the test that you're doing to determine if the animals are carrying the virus?
Yeah, thanks. So the test that we do to screen for the presence of the virus is a PCR test, so it's polymerase chain reaction. It's basically a test that amplifies a part of the viral genome or the viral RNA and looks for a conserved segment of that viral genome. And the PCR test amplifies that segment many, many times so that we can detect it. So in general, PCR tests are very sensitive and very specific for the virus that we're looking for. But they're not always perfect. And so depending on the test, there can be false negatives, more likely than false positives, but both are possible. But in general, those are both very low for PCR tests. So we're just looking for that little piece of viral RNA that indicates that the virus is actually there. I mentioned at the end that we're also looking to add serological tests now to this study to understand if there are animals that have been infected and generated antibodies to the infection. And the serological tests look for those particular antibodies that indicate that the animal mounted an immune response to an infection. And we expect that even if transmission is a very rare event, one that doesn't happen very often, even in close contact situations, that it happens more often than we're able to detect through direct sampling for the virus, because our direct sampling is only at one point in time, whereas if that animal gets infected and generates antibodies, we expect the antibodies to be there for a much longer period of time. So we can sample an animal for antibodies after they've been infected, probably a month or maybe even 3 months. I'm not sure exactly how long, but for quite a while longer, and detect the fact that that animal has been infected. So we're looking forward to adding some serology. Part of the reason why we haven't until this point is serological tests are much more difficult to develop, especially if you're looking at a variety of different host species. And serological tests have in general, have much greater problems with being less specific and less sensitive, and have to be validated in a much more rigorous way than the PCR tests.
So you mentioned the low likelihood of transmission there, Dr. Runstadler, but several people are wondering about that. And what's the risk of one dog passing it to another, or a cat passing it to another cat? And of course, wondering about the risk of transmitting from a pet back to the humans, and whether they could pick it up from the fur or not, as one right. potential route. And somewhat related to that is, whether or not the animals will necessarily show any particular symptoms. So if it was possible right. to come back, would they know without a test that their animal was infected? So I'll sort of a full range there, and I'll let you speak to those.
Yeah. So those are all, you know, obviously questions that I think many of us that have animals are concerned about to some extent, and it's a big group of questions that are all interrelated. There, you know, I should say there's no definitive answer to those questions, but I can tell you what we understand now about the virus. With the caveat that this is based only on the evidence that we have, of course, and that's one of the reasons why we're doing the study that we're doing, is to generate some data and some evidence to support some of these assertions, I guess. So it does appear that based on our data and that of others, and based on the fact that we've only seen a few documented cases of transmission of virus from humans back into animals, or into animals that are in close contact, that this is a rare event. What we don't know so well is perhaps how rare and what sort of circumstances make that event happen when it does. And I guess that's really the details of the heart of the matter there that we'd like to know, and we just don't, right? Because those events where transmission has occurred have been too rare and not well enough documented to really understand in a scientific way why they occurred. But what we can take from that is that transmission is possible from humans into pets. So a lot of people are worried then about, well, am I going to infect my pet, or is my pet going to infect me? So the best advice, based on our current understanding that event could occur, is that you treat your pet in a similar way to ways that you might treat a human that you are in close contact with. So if you are COVID positive, or you suspect that you've been in contact with somebody that's COVID positive, that you practice social distancing in a way with your pet that you might with a family member. It's likely that possibility is rare, that you would transmit into the pet. And there's no evidence currently that pets have been involved in transmitting the virus back to a human host. And there's several reasons that might be. One might be behaviorally, that just doesn't happen, that the types of contact are not conducive to transmitting the virus from the pet back into the human host. It might also be that virus, when it moves into a pet, undergoes some changes that make it less likely to move back into the human host. But either way, the rare event of transmission from a human to a pet is not matched, at least currently, from the data by transmission from the pet back into the human host. Even so, we wonder whether our pets might be fomites, or a vehicle by which virus can be transmitted from one person to another person. So there's lots of questions that we've gotten about, can virus be deposited on the fur of a pet, and you pick it up by petting that pet? So there, again, there's not real definitive answers, but there's some data that we can go on a little bit to understand how likely that may be to occur. There have been a few studies looking at survival of the SARS virus on surfaces, and the virus does appear to be able to survive on some types of surfaces for hours and possibly even days. But in those studies, the surfaces that virus survives on tend to be inorganic, smooth surfaces like metals or plastics. And on surfaces that are more organic, like cardboard or cotton surfaces, the virus doesn't survive nearly as long. And that's probably because on organic surfaces, the virus is bound up more on those surfaces or even inactivated by some of the characteristics of those surfaces. And so we might put pet fur or the surface of an animal into that organic context and say that it's unlikely that virus will survive for long periods of time on pet fur, but still allow at this point that it might be that it's a possibility. And as you can imagine, doing studies on survival of a dangerous virus on animal fur are very difficult logistically, and so that's in part why those studies haven't been done yet. But from the data that we have, I think we could infer that that's also a very low likelihood. But again, the possibility exists. So probably the best advice in this case is still to maintain social distancing with your pets and other pets that aren't part of your family. But realize that the opportunity and chance of transmission in those cases is likely also low, and if not absent.
So there are several questions, Dr. Runstadler, about what people could do, and you've kind of alluded to a number of those there. But maybe if I may, I'll just say a couple of minutes what we do here in the Hospital for Small Animals, because there are some questions from people who were dog groomers. Do they need to wear gloves and a mask to protect themselves? What can they do with their pets? And so, as with most veterinary clinics, and I'm sure many of you have either had this experience yourself or have heard about it, we're much more concerned with risk for our staff, so our veterinarians and our veterinary technicians, from the owners, and much less from the animals. So we always ask the owners whether or not they have COVID-19, if they've been diagnosed with it, in which case we ask them to have somebody else bring the animal in, or if they've had any flu-like symptoms or signs or anything that could be consistent with COVID-19. If they don't, we don't do anything special with the animals. We are all wearing masks inside the clinic, but that's to protect ourselves from each other, not from the animals. And we don't wear gloves when we handle the animals. If we do have an animal from a household that has COVID-19, or the owner has COVID-19, and this happens on a relatively regular basis, we are a bit more cautious, and we will wipe their fur down with just alcohol wipes. We don't use any disinfectants. You don't want to be spraying them. You don't want to soak them. And if there was something on the animals, some people have suggested you could bathe them, but if there were viral particles on the fur, then that might just aerosolize them. So all we do is we do a light wipe down with an alcohol wipe, and honestly, as you said, Dr. Runstadler, we think the risk is very low, but that's the only specific care we take with the animals. From studies, we know that dogs don't seem to transmit it to each other. Cats can transmit it to other cats, but the majority of them don't show any clinical signs.
I think that's an important point, that in the experimental studies that have been done, the infected animals, whether they're dogs or cats, don't show clinical signs of infection generally. And as you said, cats seem to be able to actually fairly easily transmit the virus from one cat to another, but that's not the case with dogs, and it's not the case with some of the other species.
And it's that fact that they don't show signs that we just treat them all the same if they come from a COVID-19 household. But we put it under what we would call the abundance of caution principle, as you've so nicely outlined there.
Yeah.
We only have a few minutes left, so let me just have a quick look through. I think we've answered all the questions on there. So let me just scroll through. What about a suggestion that the animals that are actually getting infected may have a compromised immune system already, and that might have contributed to them being susceptible.
Yeah, it's a good question, and it's definitely a good question for human patients as well. You know, we and other groups certainly are studying the same kinds of questions in the human population as we are in some of our pets. So we've, as a part of our study, we've also included sampling animals, for instance, that are coming in for treatment for cancer, and they're either immunocompromised because of some of the treatment that they're undergoing or because of some of the disease conditions. And so we're really interested in those situations. You would expect that an animal that's immunocompromised might be more susceptible to infection and perhaps to showing signs of disease if they are infected. But again, there just doesn't exist at this point. You know, it's hard to believe. It seems like it's been so long, but it's only been a few months that this virus has been circulating. So we don't have that data at this point, but it's a hypothesis that we'd like to be able to test. And I think from an intuitive standpoint, if you're in that situation, you have an immunocompromised pet, that would be one more reason to be cautious and take an abundance of caution, particularly if you are diagnosed with COVID-19 or you have a close contact who's diagnosed with COVID-19.
So there's a number of questions here that relate to transmission, and I'll kind of summarize them. You know, the first one is what's the likely route of transmission of both, I think, giving it to the animals from people, or if an animal has it and did pass on to another animal. You know, is it aerosol or not? It's a question about whether or not ticks or mosquitoes could perhaps bite one animal and transmit it to another. And somebody was concerned about it had been suggested by their veterinarian that they not bathe the pet or put anything on the pet because it could drive the virus down to the skin, and then that might increase risk. So I think that's really asking the question about transmission through the skin. So those are sort of related questions. I'll turn it over.
Right. Yeah. The virus doesn't. This is not a virus that seems to be transmitted through a vector or an insect, a biting insect or tick. Again, there's no evidence that that's the case, and that the virus could survive that type of transmission. Viruses that do that are very well adapted for doing so, and this is not a family of viruses that is associated with ticks or biting insects in general. So the questions about transmission are also good ones, again, for which we wish we had more data at the current time. The abundance of data really shows that this is likely mostly an aerosol transmitted virus in the human population, or by droplets or aerosols that are coming out of the respiratory tract. And that's likely the route of infection and transmission in those cases where it's been documented to a pet. It's a very close contact for a long enough period of time that the pet animal got an infectious dose of virus from an infected human host. It's interesting, though, that this virus, excuse me, and other coronaviruses also have the capacity to infect cells in the digestive tract. And in cats, it has been shown in experimental infections that cats shed a lot of virus in fecal matter or through the digestive tract. But it's not clear in the cases where those experiments have been done whether transmission between cats is through aerosol transmission and the respiratory route, just like in humans, or whether it might actually be through fecal oral transmission and a shared litter box, perhaps in this case. But either way, again, that doesn't seem to be a big risk for human health. But again, as with cleaning a litter box, in any case, it's a reason to be careful when doing it.
So there are many, many more questions, as you can imagine, Dr. Runstadler, but we are past the time when we need to wrap up. So I'd like to thank you for an excellent presentation, for answering all those questions so well. For those of you whose questions we weren't able to answer, we apologize. They're all great questions, and they're still coming in as you've been answering them, but unfortunately, we just don't have any more time. So with that, I think we will conclude today's presentation, and thank you very much everyone for listening, and thank you again Dr. Runstadler for the research that you're doing, which is ongoing, and you're still recruiting patients. And
we are, yeah.
You can check out your website for more information, correct?
Please do. It's sites.tufts.edu/covers. And there's a form on there where people can indicate their interest in participating in the study. And again, we're particularly interested in enrolling people in the home study who are in more of a high-risk situation. But please go to the website and look at what we're doing, and hopefully some of the information that we have on there can answer additionally some of the questions that people have.
Okay, well, thank you very much.
You're welcome. Thanks everybody for tuning in.